Test, test. Un, deux. Un, deux. Salut, salut, salut. Un, deux, un, deux. Un, deux. Un, deux. Hi, everyone. We will begin shortly. Okay. Well, good morning, everyone. I hope, thank you for joining us for the second day of conference. I hope you enjoyed the first breakfast and the cooler hours of the morning. Today, our opening session will be an animal behavior session. And to kick things off, I'm honored to introduce our keynote speaker, Dr. Paul Tixier. So, I will say a few words about Paul. Paul is a behavioral ecologist whose research focuses on marine mammals, on their interaction with human activities, particularly in the Indian Oceans and Southern Oceans. After completing a master's degree in marine ecology in the University of Victoria, British Columbia in Canada, and Aix-Marseille University, and spending summer doing fieldwork on killer whales on the northern Vancouver Island, he completed in 2012 a PhD at CNRS in the Centre d'études biologiques de Chizé on the interaction between killer whales, sperm whales, and fisheries on the Crozet subantarctic islands. While still working on these populations, he extended his research to include fisheries interaction with other marine predators, especially marine mammals, in Australia, the Indian Ocean, and other subantarctic islands through the subsequent postdoc he did first with CNRS, then the Deakin University in Australia. In 2021, I think, yes, he was appointed as a researcher in the French National Research Institute for Sustainable Development, which name is IRD, and joined the Marine Biodiversity Exploitation and Conservation, which is Marbec. It's near here in the city of Sète. So his research now focuses broadly on behavioral ecology of large marine predators and human wildlife in oceans. In particular, much of his efforts are dedicated to understand the behaviors of marine mammals and the drivers of their direct and indirect interaction with fisheries. For instance, he authored in 2021 a global review on depredation by large marine predators on fishery catches, highlighting the limitation of current deterrent methods and the need for interdisciplinary approaches to address the growing challenge. From this knowledge, he also uses these interdisciplinary approaches to identify ecologically and socio-economically sustainable way to reduce this conflict associated with interactions through changes in fishing behavior and also fishery management. So this is precisely the focus of his talk today, of the session. So I will now step aside and let Paul take the stage with his talk, understanding the ecology of behavior of killer whales in the southern, just with fisheries.
Thank you, Pablo. Yeah, thank you very much. Well, good morning, everyone. Sorry, I'm a little bit sweaty. I came by bike and it's already very hot out there, so I'm gonna try to cool down. So yeah, I changed the title a little bit, making it a little bit broader. But the idea is for me today to present some of the findings we had with my students and collaborators on the ecology and behavior of a killer whale population interacting with fisheries. So to begin, just to put things into context. Oh, yeah. In the marine environment, we are in the context of an expansion of human activities and especially fisheries that is pretty recent. Fisheries have expanded worldwide over the past 50, 60, 70 years. This is what you see on this graph on the world fishing catch. And basically today, fishing activities are pretty much everywhere in all oceans and in all seas of the planet. And in a report in 2020, the FAO indicated that nearly 1/3 of the fish stocks of the world are overfished. And with this expansion of fisheries in the world, there's been an expansion of conflicts between this human activity and large marine predators, marine megafauna, such as seabirds, sharks, and marine mammals, most of these species having vulnerable conservation status. And those conflicts, they translate in indirect interactions, everything related to competition for resources between fisheries and these species, and indirect interactions. The two main ones being bycatch, those species, when they come into contact with fishing gear, they become accidentally taken, and the other one is called depredation. So depredation is actually a behavior that many species have developed, essentially toothed whales, odontocetes, pinnipeds, and sharks. And this is what they do when they come and feed on fish that is caught by fishermen on fishing gear. So you've got different instances here showing pinnipeds taking fish from fishing nets, dolphins around nets, killer whales around long lines, and sharks on tuna. And so this behavior has been increasingly reported in the world over the past 40 years. We've got many fisheries across the world that have documented depredation by large marine predators. And it's actually in all fishing sectors, industrial, artisanal, recreational, and it involved an increasing number of large marine predators from marine mammals and sharks. In this review we published in 2021, we actually documented more than 200 fisheries in which depredation had been documented, involving nearly 70 species of shark and marine mammals. The problem with depredation is that it causes losses for the fishing activities, but also involve risk for these species. And we found that nearly 80% of those species were caught or killed by fishermen when doing depredation around the fishing gear. So there's been an effort to find solutions to these conflicts resulting from depredation over the past 50 years. And initially, the main approach that was used was lethal control. Basically, fishermen, even governments, were actually killing these species seen as competitors or pests by the fishermen. And since the 1990s, it's more an approach of coexistence that is being tried to find solutions to depredation. The fact is, today, solutions to these conflicts remain limited. There's been a number of attempts pretty much everywhere in the world, with first what we call the technological approach. So basically, fishermen are trying catch protection devices. They're trying to repel the marine predators using acoustic deterrence, for example, or to change the fishing gear. Another approach to try to mitigate the issue is what we call the behavioral approach, is how fishermen can actually change their fishing practice to avoid those interactions or to minimize the amount of fish that the marine predators take on the fishing gear. The problem is most of these approaches don't work because there's a big gap of knowledge essentially on how depredation works, the mechanisms of depredation, which are the causes and consequences of this behavior as part of the conflict. When it comes to understanding the drivers of depredation, sorry, that slide is pretty heavy, but if we place yourself into the framework of prey switching to anthropogenic food subsidies, what drives those species to change their behavior to feed on food coming from humans? Well, there's a number of factors actually that are involved. Some of them are associated with, I think I've got a pointer here. Yeah, with the food itself, the nutritional value, the facilitated access. Some of them come from opportunities. For example, how much the range of the species overlap with the human activities. And a lot of them come from the wild species, so their biology, their behavior, their ecology, the environment they live in, and the habitat and natural resources availability, for example. Of course, all of these are influenced by humans as well. There's a whole component of these interactions involving human behavior, perceptions, and attitudes as well. But in this talk, I'm going to talk to you about that component, essentially, the ecology component, which has been the focus of part of my research over the past 10 years. How about the consequences of depredation? So obviously, large marine predators feeding on fishing gear will cause socioeconomic impacts for the fishing activities. Fishermen are losing fish. Sometimes there's damage on the gear, but it comes from all the extra effort they need to provide to compensate the losses. There are costs for the fish stocks as well. It's really hard very often to estimate the amount of fish that is taken by the marine predators on the fishing gear, which causes uncertainty in fish stock assessments. And then you've got impacts on the predators and their ecosystems. So these impacts can be negative. They can be caught on the fishing gear, for example. Fishermen are trying to kill these species. that they see as competitors, but you also have negative and positive effects associated with anthropogenic provisioning effects. So these effects come from the opportunity for these predators to feed at low foraging effort. On one hand, it can increase the fitness of individuals, enhance populations, but also it has the effect of changing potentially the diet of these animals by providing them with an easy food. And all these effects combined can potentially lead to a modified role of these species as top predators in ecosystems. And there's two papers that are super important that I strongly recommend that actually looked at these effects in both terrestrial and marine environments. That one from Oro et al. And the other one from Newsom, looking at the effects of resource subsidies on predators. As part of my research, and what I'm going to present to you today are three main questions we've been trying to address. The first one is who is actually depredating? How many individuals in populations of large marine predators are involved in this behavior? Is that a subset of populations, or is that the entire population that has developed the behavior? The second question, it's what drives it, and especially what aspects of the ecology of individuals can contribute to engaging in this behavior, and what is the role of prey preferences. And the last one is how does it impact populations? What are the effects of depredation on the demographic performances of individuals, but also on populations? And these questions, we've been trying to address them using one species in particular, which is the killer whale. This is a typical picture of a group of killer whales cruising in Canada, for example. And just as a reminder for those who don't know that species very well, it is a dolphin. It is the largest member of the dolphin family. It's pretty much everywhere in the world, essentially in cold waters, but you do have killer whales in tropical hot waters as well. It's a pretty big animal. There's a strong sexual dimorphism between males and females. Males are bigger, but females live much longer. Females can live up to 90, 100 years, and they've got a post-reproductive lifespan. It's a highly social species. They live in what we call a matrilineal social system. But dispersal patterns vary between populations. We've got different systems of social organizations according to populations. Same with the feeding ecology. As a species, killer whales can eat pretty much everything. But when you look at the population level, there's strong levels of specialization, in the prey preferences differences. In the world, this variation in the feeding ecology between populations have led people to characterize what we call different forms of killer whales, what we call different ecotypes of killer whales. And these ecotypes are genetically different, morphologically different as well. And they are defined as populations that have totally different feeding ecology and are not breeding with each other. For example, in Canada, we've got two ecotypes of killer whales living in the same environment. At the top left there, we've got the resident killer whales. They are fish-eating killer whales, and they share the same environment with what we call the big killer whales in the middle, which are marine mammal-eating killer whales. And what's striking is that these two populations of killer whales use the same habitat, but a different niche. And the big killer whales are actually closer to a population that is in the Atlantic genetically than with the resident killer whales that are living in the same area. In the Antarctic, we've got multiple ecotypes, some marine mammal eating, that is the top right here, with the type C in the Antarctic that are fish-eating killer whales. In Norway, we've got generalis killer whales that are a mixed diet of fish and marine mammals. And in Patagonia, we've got marine mammal eating killer whales. The killer whale is actually one of the species most frequently documented depredating on fish caught by fishers on fishing gear. So having developed that depredation behavior. In the review we published, there are nearly 30 fisheries with killer whale depredation documented around the world. And when you look at the map, this is how it goes. You've got killer whale depredation in Alaska, in Russia, in the Northern Atlantic, and in the Southern Hemisphere in subantarctic waters. And the main fishery these killer whales target is what we call longline fisheries. Longline fisheries are a fishing technique that is using lines with thousands of hooks, each hook being baited. Fishermen leave the hooks in the water for hours before they retrieve the long line, and this is when the killer whales come. And on that video I'm showing now, the fishermen are actually retrieving a long line, and killer whales are removing the fish one by one from the hooks. They're pretty good at it. They usually don't get caught or injured by the fishing gear. They take gently the fish and remove it just by pulling on it from the hook. Killer whales are, as I say, pretty good at it. They can actually remove large amounts of fish in those fisheries. For example, in Alaska, they remove nearly 400 tons of sablefish every year. In the Southern Hemisphere, it's 317 tons of toothfish every year. And in the Southern Hemisphere, we described that multiple populations and even forms, ecotypes of killer whales, have all developed the depredation behavior independently. We've got at least 3 forms of killer whales from southern Chile to southern Australia that have developed the behavior pretty much at the same time. Today, I'm going to focus on one population from the southern Indian Ocean that we've been studying, and especially since I did my PhD in Chile. So this population is around the Crozet Islands, which is part of the French subantarctic islands in the southern Indian Ocean of the Southern Ocean. There are 5 main islands in this archipelago, the main one being Possession Island here. Some of you in the room today have been there as part of fieldwork. This is how it looks like. We've got lots of animals, essentially marine mammals and seabirds in there. And around those islands, we actually describe 2 forms of killer whales. On one hand, the ecotype that we call the Crozet ecotype, pretty classic looking killer whale. And on the right, we've got what we call the type D killer whales. This is a new form of killer whales that was described only recently. That was about 12 years ago that the first paper came out. These killer whales are very mysterious, but they are very weird looking. They've got a very tiny eye patch, which makes them very different than any other killer whales in the world. Obviously, we knew the type Crozet before the type Ds, and that's because these killer whales have both an offshore and inshore distribution. They are the killer whales that we see most often, and they are subject to most research effort. The other killer whale, the type Ds, we don't know anything about them. They live far offshore. We don't know what they're eating, what the ecology, but they also interact with fisheries. So let's focus on the type Crozet killer whales. So this is a map showing the distribution of encounters of killer whales around the archipelago. So you've got sightings from the coast. This is the picture here in the right-hand side. Killer whales can actually come very close to the shore around the islands in this area. And then you've got a picture of offshore encounter where you see the ocean is much rougher than in inshore waters. This population has actually been subject to a long-term monitoring program, and they've been monitored using a technique that we call photo-identification. So basically, this technique was discovered in the 1970s. It's possible to identify each individual by natural marking on the dorsal fin and the saddle patch, that patch that is right behind the dorsal fin, and also the eye patch. So each individual has unique shape or markings on these characters. And all together with this data, by taking pictures, a lot of pictures of these killer whales, we can reconstruct what we call the photo identification catalog and monitor these individuals through time. The first pictures that we received were actually back in the 60s. We managed to identify killer whales back in the days when people were taking photographs when they would see killer whales. But the first dedicated study happened in the late 80s, early 90s, by Christophe Guinet, who was my PhD supervisor. He's a researcher at the Centre d'études biologiques de Chizé. And this is how it looked like. Those killer whales were not afraid of humans. They would come very close to shore, and people would actually interact with those killer whales on a regular basis around the island. And so Christophe, in this first dedicated study, actually showed that these killer whales were quite different than most of the other killer whale populations in the world. They were feeding on pretty much everything. They were very opportunistic. They were able to feed on large whales, on seals, on penguins, but also on fish. And for that, they've developed a number of hunting techniques that are highly different from one situation to the next. And altogether, this characterized a pretty unique culture for these crozet killer whales. So for example, they feed on southern elephant seal. It's one of the main prey of this population. And to catch them, they've developed 3 different techniques. The first one is the most impressive one. Basically, killer whales intentionally strand on the beach to catch elephant seal, and especially the pups, right on the shore before coming back to the ocean. This is a high risk hunting technique, requires long apprenticeship, and it's a technique that is potentially lost today. I'll come back to that later. Killer whales are also able to catch seals along the rocks, along the shoals, along the beach, but also in open waters in the bays around the island. The other prey species are penguins, 2 species, king and gentoo penguins, that they catch opportunistically around the rocks and the kelp forest. But further offshore, they're also able to take down large whales. And this is a picture that was taken in 2013 of the same group of killer whales that we will see around the island going after southern right whales. In the first dedicated study, Christophe also described these individuals of the crozet type population as associating in very strong, stable groups, which were likely to be matriline's groups. But in the 80s, he also noticed that this population had very low reproduction rates, very low numbers of cows per reproductive females. In fact, this population was exposed to a major change in the environment in the 1990s. So in 1996, a commercial fishery started in the area targeting this fish species, the Patagonian toothfish. This is a big fish, can be up to 2 meters, can be 100 kilograms in weight. And right now, the fishery is small. There are only 8 vessels that are allowed to fish, but they fish quite a lot of toothfish. So they use those baited long lines with 3,000 to 600 hooks per line. They've got vessels that are up to 60 meters in length. They all come from the Réunion Island. They go at sea for 3 months, and they are managed by the TAF administration with fish stock assessment by the Museum of Paris. This fishery is highly regulated. All fishing vessels, every time they go at sea, have fishery observers that monitor all fishing activities. And right now, the quota is about 6,000 tons, for crozet and kerguelen, and about 1,000 tons just for crozet. The problem is, when the fishery started in 1996, along with those licensed fishing vessels, many unlicensed fishing vessels came into the area. So what we call illegal fishing vessels that were actually working around the archipelago, and you could see dozens of fishing vessels around the Crozet Islands at the time. And in that same year, fishermen from the licensed fishing vessels reported killer whale starting depredating on fish right away. As soon as the first vessels got into the area, killer whales started to understand that they could actually feed on the fish taken on the long lines by these vessels. So this is how it looks like. I'm going to show you another video specifically made around the Crozet Islands with a camera that was set up on the line. And those killer whales, they just follow fishing vessels and they work in groups. And every time they can find a long line, they will remove those fish. So altogether, Crozet is actually a place where the highest depredation levels have been recorded. Killer whales are around fishing vessels for around 40% of fishing operations. And every year they remove 180 tons of fish, which is worth about $4 million for the fishing sector. And it's about 20% of the total catch, which is up to 30% when you put sperm whale into the equation. Sperm whale is another species that has developed depredation as well in there. And as you saw in the video, sometimes they take the fish without even eating it, which gets the fishermen very upset. So with the arrival of these fishing vessels, we extended the photo identification monitoring program, not just to the island where it was previously, but to the fishing vessels. So from the fishing vessels, we implemented protocols for the fishery observers to take photographs of killer whales when they were depredating around the fishing boats. And we also extended the effort to other fisheries, just like the one around Prince Edward and Marion Island. Sorry, I'm going to drink a little before I get into the findings. So now, coming back to the questions we've been working on, I had a student, Morgan. She tried to address how many killer whales are actually involved in depredation, have learned that behavior as a new foraging strategy. And to do that, she used that photo identification dataset long-term, and she applied macro-capture models to this data. And what she found out, but estimating the number every year, is that in the early years of the fishery, when the program started, there were not that many killer whales that were involved in depredation, around between 30 and 40. But that number increased over the years until 2014, when 100% of the killer whales of the population had developed the behavior. So the findings suggest that it actually took about 18 years since the start of the fishery in 1996 for the new behavior, depredation, to spread across all the individuals of the population. With the photo identification dataset, we actually focused our efforts into 23 social groups that we see pretty much every year and that are responsible for most of the depredation events. Basically, these 23 groups are the 98% of all killer whales that are involved in depredation in the area. And I had another student who is actually defending his PhD thesis in September. He's pretty stressed at the moment, but he worked on the first question is how those killer whales have learned depredation. So using the same data set, he first analyzed the social organization of the population by looking at the social strength, so the social affinity between these different groups. And he looked at the effect of social affinity on the probability of these killer whale groups to learn the behavior. And what he found out is two learning pathways actually occurred. On one hand, some groups developed the behavior through social learning by associating with killer whale groups that had already learned the behavior through horizontal transmission pathways. But on the other hand, he also found out that some killer whale groups developed the behavior without interacting with other groups, showing independent learning. Erwan had another study looking at, all right, we've got these 23 groups involved in depredation, but do they do depredation to the same extent, or are there variation across the different groups within the population? Well, he actually found that there is a strong heterogeneity in that depredation behavior that is first translated in the probability of having those social groups of killer whales around fishing vessels. Some groups are actually responsible. Well, we've got 4 groups here. If you see the pointer, those four groups, they are responsible for nearly 80% of all depredation events. We've got 4 groups that become highly specialist into the depredation behavior. And that variation in behavior translated also into the area over which they interact with fishing vessels. Some groups are able to find and to follow fishing vessels pretty much everywhere around the fishing area, while other groups always interact in the same small specific area. Going into the second question, which was a major question mark for years, is toothfish already part of the diet of these killer whales? Or are fisheries bringing toothfish as a new prey by facilitating access and taking fish from the bottom? Well, just to give you a little bit of context, toothfish is quite a special species. There's not much known about it, although it's the primary economic commercial fish in the Southern Ocean. But this is a picture showing you how big it can be, and it's essentially a bottom fish that can live pretty deep. But the juveniles stay in shallow waters. Small adults go a little bit deeper, around 500 meters, and the large adults are usually around 1,000 meters. So very deep for a killer whale. Killer whales can dive up to 1,000 meters. This is the world record, but usually they don't go any further than 400, 500 meters. So large toothfish might be a hard prey to catch in natural conditions without any fishing vessels. So how do we assess or quantify the extent to which toothfish is part of the diet of these animals? Unlike for whales or penguins or seals, it is really hard for us to actually confirm that killer whale are actually taken a toothfish by direct observation. So what we use is the indirect methods. Many of you are familiar with it. We actually collected tissue samples from killer whales and from potential prey. From these killer whales, we had two groups: killer whales that had interacted with fisheries and killer whales that had not interacted with fisheries before the sample was taken. And we compared with prey samples through an isotopic analysis, looking at carbon and nitrogen and using mixing models along with prey sample signatures. A biopsy sample, I'm going to remove the sound here. So this is from a fishing boat operating around the Crozet Islands, and basically what we're using is a crossbow with special biopsy darts that we trigger onto the animal, and that will remove a tiny piece of skin and blubber on the back of the animals. So this is how it's done. We're using a tether line to retrieve the arrow and get the sample back on the ship, which you're going to see here, and a very happy person. You see the sample. There you go. So this is from fishing vessels and from the shore of Possession Island, where we also run some dedicated work. So as I mentioned earlier, those killer whales can get pretty close to the shore. They react very strongly to noise that we make when we walk along the shoreline. This is actually how they catch elephant seals. And that day, they were practicing. We had a number of young killer whales that were practicing stranding. So you see the weather is not that good, but I was trying to load a second boat, trying to get the killer whales to stay with me. And I wanted to get, sorry, it's moving a bit, that individual at the back, which at the time was a 3-year-old female. And so this is what they do. For hours, they will practice stranding. And this is the mother. I had already got a sample from her. And basically, I was just waiting for that individual to show enough area on the back to get the sample. Oh. And there you go. And so sometimes they do react. They feel something. It's powerful. They're big animals, but still, it can be painful. But most of the time, killer whales are pretty interesting because they don't react and keep doing what they do. So from these samples and from this analysis on stable isotopes, we actually got that graph to the right. So the prey samples were grouped into 4 different groups. large whales with one species of penguins, small tooth fish with elephant seals, adults, and medium-sized tooth fish with elephant seal pups, and large tooth fish. And the first finding that we got is that from the non-depredating killer whales, the ones that had not interacted with fishing vessels before sampling, well, the contribution of toothfish that we estimated from stable isotopes was pretty high, around 30% of the diet being reconstructed. So this suggested that toothfish is potentially a prey item that these killer whales take naturally without fishing vessels. And when you look at the difference between those killer whales and killer whales that had taken toothfish from longlines, the difference is not that great. It's actually a little bit higher, suggesting that potentially fishing vessels do bring a better access to those large toothfish these killer whales have to put a lot of effort to forage on in natural conditions. So fisheries may facilitate access to these large toothfish and may increase the intake of their resource by killer whales. But again, when you work with killer whales, you got to get used to the fact of working with small sample size and being very careful with your findings. So we only had 20 samples of killer whales at a time to run this analysis. Another student I had as a master's student, she looked at, all right, those killer whales are taking fish from long lines, but how much does it contribute to the energy intake of these killer whales in total compared to other prey species? So from the 180 tons those killer whales remove per year, Joanna, she actually estimated that when killer whales find a fishing vessel, usually they will fulfill 100% of their energy requirements. And when you bring that estimate at the year level, and for the whole population of killer whales, that contribution is actually much lower. It's in between 2 and 9% for all individuals for a whole year. So when depredating, killer whales are actually not doing anything else. They're just eating toothfish on the long lines. But one question remains: to what extent does this contribution replaces what killer whales would have taken naturally? So this, we don't have the answer yet. Sorry about that. Still working on it. Going into the last stage, the last question we've been working on, so about the impacts of depredation on the crozet killer whale population. Well, in 2010, there was a first demographic study that was published on the population using the photo identification data from the Possession Island. And what this study showed is that the numbers of killer whales around the island dramatically dropped during the 1990s. From a little bit more than 100, there were less than 40 killer whales that were seen in the late 1990s, early 2000s. And so people at the time, and including Christophe, were like, what's going on in this population? And what they were observing is that in those different groups of the population, they were losing members. It was not just group disappearing and doing something else. It was the same groups being observed again and again, but without some killer whales, including juveniles, including reproductive females. That makes no sense for them to disappear at the time. So they suggested that during this period, there was an additive mortality event going on. And so what we did later, and that was part of my PhD, it was to look at one specific variable, which was the effect of illegal fishing on demographic parameters, and especially apparent adult survival of the population. And for that, we also use CMR models using the full identification data. And what we showed is that before the fishery started, the survival in this population was high, was nearly 1.99, which is normal for such a species, long-lived species, and it's comparable to healthy killer whale populations. But when the fishery started, that survival dropped to a little bit more than 0.9, and especially during the illegal fishing period between 1996 and 2002. After 2002, the French government implemented surveillance activities around the island to reduce illegal fishing, and actually worked. They managed to get rid of most of the illegal fishing activity. But after 2002, while the survival of these animals didn't come back to the initial levels. It actually increased again, but stayed around 0.96, which is still lower than healthy populations. Oh, sorry, I had an animation here. So this additive mortality event was likely explained by something that happened with illegal fisheries. And as it turns out, those fishing vessels, those killer whales, couldn't make the difference between a legal and an illegal fishing boat. And those illegal fishing vessels, fishers on board were using firearms and explosives to get rid of killer whales taking the fish because the costs were just too high. And so these practices were being used for years around the Crozet Islands without people knowing about it. And we only knew about it later. But what we saw, thanks to that long-term data set, is that it did impact the killer whale survival during the time. How about now? We ran another analysis, more recent, and what's concerning is that that killer whale population is still declining. So basically, between 2005 and 2020, using these 23 groups that we see every year, we used a direct census approach to see that the numbers have dropped by 14% during that period, with more deaths than births, running the total of 87 individuals in 2003. And what was even more concerning is that mortality rates were abnormally high in juveniles and reproductive females. This is where the survival is usually the highest for this species. On the other hand, we looked at the effect of depredation on not on survival, but reproduction. And what we found out is that females, reproductive females, that interacted a lot with fishing vessels had a higher reproductive rate, so a probability of calving. So that was really hard signal to pick up. Again, we need to be careful, but basically the more they interacted with fishing vessels, we did have a significant effect of that variables, sorry. And the mean calving rate today is 0.2 calves per reproductive female per year, which is equivalent to healthy killer whale populations. So what we've got now is a demographic anomaly. Those killer whales have a low survival, but a good reproduction. And this is not normal for a long-lived species just like killer whales. First you survive and then you reproduce. Mortality rates being high for juveniles, reproductive females, that's something else. And so what we try to do is to understand the cause of that anomaly in the demographic patterns of the population. And this is especially concerning since some of the prey that these killer whales are after, and especially large whales and elephant seals, are going up. There's been a period where these two prey species were very low around the Crozet and Kerguelen Islands. But now, and since that study in 2023, you see the number of elephant seals is increasing again around the Crozet Islands. So one of the assumptions we looked at to explain that decline that is still going on is whether this additive mortality in the 1990s had an extended effect on the population through a change in the social organization. Okay, I'm going to rush because I still have quite a few slides to present. So briefly, what we showed was a positive correlation between group size and the strength of association in this population and their survival. And what happened was, before the additive mortality event, those killer whales were in stable groups with strong and high associations with others. But when there was this mortality, those surviving killer whales, they had to reassociate with other groups more loosely, more opportunistically. And this had definitely an impact on their survival. They were struggling to maintain that optimal group size they needed to survive. The other cause that we looked at is, are the killer whales around the Crozet still exposed to illegal fishing practices? Well, from the photo identification data, we actually reconstructed the movements. So these are all the encounters of killer whales linked with a line when it's the same group or same individuals being encountered. And what we found out is those killer whales, they move a lot. They actually go in international waters, and they especially go into that area between the Crozet and the Prince Edward Islands. And in this area, is known to be still subject to poorly regulated fishery, even illegal fisheries. And that was a study done using albatrosses with specific tags designed to detect fishing vessels. I'm not going to go into details, but that was a pretty neat study. That finding, plus the fact that in 2019 we had a killer whale coming back with a bullet wound in the dorsal fin, confirmed that these killer whales may still be exposed to illegal fisheries using lethal practices on them. So I'm getting to the conclusion. What do we get from this study on the crozet killer whales? Well, first, about the drivers of depredation. We did confirm that the depredated fish is part of the diet of these killer whales, and opportunities to depredate may actually arise when killer whales are foraging in the same areas as the fishing activities occur. And killer whales were already seeing tooth fish as a prey, which explains why they developed depredation as soon as the first fishing vessels arrived into the area. Second, the consequences of depredation, which have both negative effects and positive effects from provisioning, but the negative effects of whales being shot actually overcome the positive effects. And the crozet killer whales is now listed as an endangered population. The study also highlights the importance of these long-term data sets. You see that many of the findings were from these long-term data. Actually, I've got a number of students working on how we can, two slides, we can convert this this increased knowledge about the ecology of these animals into mitigation solutions. So Margot, who is doing a PhD with me in SET, from all this information about the ecology and the feeding preferences, she incorporated that into predictive models. And now she was able to actually predict where depredation was most likely to occur. I got another student, Vinicius. He's combining tagging data with fishing data to look at that overlap between fishing activities and killer whale foraging areas using satellite tags. And to look at also to what extent killer whales are actively following fishing vessels and over what distances and how they can detect them from the distance. So altogether, this type of information is actually being transferred to managers and fishers for them to implement avoidance strategies. And last slide, when looking at the bigger picture again, what keeps me awake at night still, and it's probably going to do so for the next years, but what we're trying to do is to understand the drivers, the determinants of this intra-population variation in the behavior, but also at the interpopulation level. Basically, in the world, we've got many killer whale populations, and some of them never acquired depredation, never learned depredation as a new behavior, although they do overlap with fishing activities. So we're trying to understand why some killer whale populations are doing it and why some are not doing it, and why some killer whale social groups have specialized into it and why others have remained opportunistic. And in terms of quantifying the impacts of depredation, what we're trying to do is to assess that change depredation may bring to the role of top predators for killer whales in ecosystems. As you saw, killer whales, when they're depredating, they're not doing anything else, and this may change the trophic interactions within ecosystems. And that's it for me. I want to thank, Christophe, especially, but also all the collaborators, and especially my students. I had quite a few this year, but it's a team that is all working on these questions, but also the people on the field collecting the data for us, and all the support and logistics from the different institutes and programs. Thank you very much. I hope we've got time for questions.
Thank you for the presentation. It was super interesting and super cool. I have a lot of questions, but I think I cannot ask all of them. The first one was about the type D killer whale. Is there any chance that they compete for the resources with the crozet killer whale, and that can explain the decrease
I don't know if the type D has the same behavior of depredation, but do they like interact with this same behavior and like compete, or even with sperm whale, for example?
Yeah, it's a good question. So the two types of killer whales, they do interact with fishing vessels. And sometimes, it doesn't happen very often, but around the same fishing vessels, you will have the two forms of killer whales. And you know it right away because these killer whales avoid each other. So usually they're all spread out around the fishing vessel and doing their things. But when the other type is there, they group up and stay close to each other. In terms of competition, they probably do compete for toothfish during those interaction events, that's for sure. So do the sperm whales. So basically, you've got to imagine that toothfish on a fishing line, it's like a zebra carcass in Africa. You've got a limited amount of food for a short time, and those killer whales and sperm whales have to rehab. In absence of fishing vessels, we don't know. We don't know how much competition interaction there is between the two forms. The thing is, they use different niches, actually. Type D killer whales seem to be using offshore seamounts, whether the closer killer whales stay on the shelf and around the island. Yep.
Thank you very much for your talk. I really enjoyed it. I have two very small questions. So one is a kind of curiosity question. Do they learn not to eat the bait and to learn to wait that a bigger fish comes? Like, does it happen that they just eat the small bait and then there's no big fish for them? And do they need to learn that? Is that why it takes time for them to learn? And the second question is, in your isotopic analysis, how can you exclude the fact that it could be another bottom feeder? So another species of fish with the same isotopic signature, and therefore that in fact they never eat this fish outside the line, they eat something else and they substitute this something else with the fish that are caught on the line. Thank you.
Yeah, so thank you. So about the first question, actually the two questions are related. Because what we observe when killer whales are feeding on long lines, they're very picky. They're very selective. They will take only toothfish because those long lines, they catch other fish species. We've got at least 3 bycatch species, sometimes large amounts of these fish species are on the lines, but killer whales don't go for it. They just go for toothfish. And so same goes with the bait. They don't go with the bait. They just wait for toothfish from the beginning. Yep.
Good. Thank you for the presentation. It was super interesting. I was wondering about this depredation behavior because fishing has existed for like forever. Basically, humans started to fish, and I did not really understand why all of a sudden this depredation behavior was so abnormal compared to what could happen before. Is it related to the industrial practices really that created this type of intense food patch, like a high dense food patch, or?
Yeah, you are right. Depredation has always existed, and there are cases back 2,000 years ago of monk seals, for example, in the Mediterranean Sea, taking fish from coastal fishermen. But it has never been much of a problem until there were less fish in the oceans. So basically, fishermen and or managing bodies, they started documenting the thing when it became a problem. And it became a problem when fishermen were struggling to keep their activity viable in time. And that was because most of the fish stocks were collapsing. That decrease in fish stocks may also explain, it's part of those factors that may drive the development of depredation and make it higher, more severe. And actually, in some areas, it is likely to occur. We see those species, and especially bottlenose dolphins, for example, increasing the rate at which they interact with fisheries. And this is correlated with the amount of fish that is available for them. So basically, they tend to switch to depredation when prey becomes scarcer. The other factor is with the expansion of fisheries, we increased opportunities like crazy. Fisheries used to be coastal, very small scale, and now we expanded the range, expanded the amount of fishing vessels around, and that had created more opportunities for the animals to develop depredation.
Thank you. Just a short following up question. So are the socio-economical costs the same for all fishing practices, or it's more important for certain fishing practices?
Oh, it's very different. There's a lot of variation. Like, for example, when you add into the equation damage to the fishing gear, and that is the case not too far away from here, actually, in the Mediterranean Sea, bottlenose dolphins on net fisheries, they take fish, but they also tear the nets when they do so. So fishermen are losing fish plus losing the gear, and the cost for them are huge. I talked to you about a big industrial fishery where the margin are pretty large, actually. They're losing 4 million worth of fish every year, euros, sorry, but they're making 60 million a year, those 8 fishing vessels. So it's fine, although it's big. But in other fisheries, depredation is definitely threatening the viability of the activity.
So it's like industrial fishing techniques are depleting the stock, creating the shortage of fish and affecting more actually the more traditional fishery, which will bear the cost basically of.
Yeah, it's a shortcut, but you can say it that way. In Alaska, for example, industrial fishing, lots of fishing vessels, but depredation has been so high by killer whales and sperm whales that they had to stop the activity and switch to a different technique.
Hi, it's working. Thanks for the great talk. It's a very naive question. I was wondering, so if I remember well, you were saying that actually the negative costs of depredation was stronger because of the lethal actions than the advantages of doing depredation. And you were saying that some groups of killer whales were not doing depredation. So are the demographic tendencies very different between those groups of whales?
Yeah, so we tried to detect those differences in that first study. And what I didn't show you because it was bringing too many details, but along with those depredating, this depredating subset of the population in the 1990s and 2000s, the non-depredating subset of the population was not doing so good either. So as you remember, I mentioned the reproduction was already very low in the 1980s. And this is probably explained by some of the main prey of these animals. And we actually underestimated the effect of whaling on this population, because now we're seeing those large whales coming back. They had an effect on the non-depredating part of the population as well. So it's a combination of the two factors that explain why the population is still declining today because of the history of low prey depletion. So demographically, they are all not doing so good. And for this period, the non-depredating and depredating animals had low survival rate as well. No other? Okay. Well,
Okay.
thank you.
Thanks everyone, and thanks again for this super interesting talk.
Thank you for the invitation.
Well, I'm sorry we had to cut short the questions, but Paul is staying here. So if you have any other questions at the coffee break or at lunch, go and talk to him. Ouais, j'attends un peu. Well, for the first talk of the day, we will welcome Aliénor Stahl, or Stoll, excuse the pronunciation. She is a PhD student at the University of Quebec Trois-Rivières on the Department of the Science of the Environment, and you will talk about behavioral thermoregulations and fine-scale movement on the Brookshire, especially the impact of temperatures on epilimnion forays. The scene is yours.
Thank you. So I'm actually a postdoc, but doesn't really matter. No problem. So we're gonna switch. We're not gonna be in the ocean anymore, but we're staying in the water. We're gonna be going into lakes today. So what happens in lakes during the summer, such as this summer, where it's abominably hot, is that the water stops being mixed and it becomes layered. And so basically we're gonna have a top layer called the epilimnion, which has a very high availability of prey, but of course has a constraining temperature. So fish will not be able to stay very long in those layers, otherwise they're gonna be, well, pretty much dying. The middle layer, called the metalimnion in green, has a lower prey availability, meaning that if you want to survive, you will need to go to the epilimnion to feed, but it is a thermal refuge, so the fish can stay there for pretty much as long as they want. And finally, we're going to have the epilimnion at the bottom, which has the lowest temperature. We don't quite know what to expect in terms of prey availability in most lakes, but we do know that O2 is a limiting aspect, meaning that the fish will not be able to stay there very long. And so a basic day in the life of a fish in a layered lake is that they're going to be spending a lot of time in the metalimnion. They will feed in the epilimnion and then go back to the metalimnion. We do know that they go from time to time into the epilimnion, to the bottom layer, but we don't know much about those forays. And so for my research since January, I've been looking into quantifying the usage of the epilimnion and epilimnion, so those bottom layers that are limited either by oxygen or temperature. And our expectation was that as temperature rises, the fish will limit the time they spend or the number of time they go into the top layer because of thermal stress, while their usage of the bottom layer, the epilimnion, would be more related to the low oxygen availability, but that could also be driven by the temperature of the epilimnion. So basically, we didn't know what kind of pattern to expect. And then the second question that I'm going to bring to you today is about the pattern. So when during the day do they go into those different layers, with the assumption that they would go into the epilimnion at dusk when the food becomes available. And for the epilimnion foray, we weren't quite sure if they were using that layer as a thermal refuge, where we would assume then that they would go during the hottest period of the day, or if it would be related to food availability, or variation in O2, which would basically change the whole patterns. And for my whole research, which is actually currently under review in the Journal of Animal Behavior, I believe, no, Journal of Animal Ecology, we also looked into the relationship between behaviors and morphotypes, so whether our fish had different traits. But for right now, I don't have the time to look to tell you about it, so I'm inviting you to come to my poster later today. So for the study system, we were set up close to Montreal, the Lake Ledoux, as you can see here, which is a rather small lake with an interesting setup, such as that we have this, oh my God, okay. We have this smaller bassin here, which is basically all epilimnion all summer long. And so we set up hydrophones, a telemetric array inside the lake for the summer 2017 and 2018. And then we tagged a total of 42 brookshires so that we were able to see where they were in the lakes in terms of latitude, longitude, but also depth. And we also were able to record their temperature, so internal temperature and external temperature, so the temperature in the different layers of the lake. This allowed us to have this kind of data, which I hope you find this pretty cool, because I do. So on the right side, you're going to have the map of the lake that allows you to see the different positions. So throughout the day, for every single hour, you're going to have the position of the fish. So this is just for one single fish. I would like to highlight this. And then on the left side, you're going to be able to see at which depth the fish is on the y-axis. And on the x-axis, you're going to have the temperature of the fish. So what you can see is that basically the higher in terms of depth the fish is, the hotter the fish is, because the fish is going to thermoregulate with the water. It's going to be basically at the temperature of the water around it. And the two red lines that you can see are basically the temperature and depth of the epilimnion, meaning that every time that fish is above the depth or temperature line, it basically means that it is doing a foray, it is out to feed. And so we transform all of that data into summaries to be able to look at how often our fish are going into the epilimnion. So here you're going to have the date on the x-axis, and the y-axis is going to represent the number of times in a day that the fish is going to enter the epilimnion. And the color represents how hot it is in the epilimnion. And what we can see is that when it's hot, our fish are not really going very often. They're going on average 20 to 30 times into that layer. But when it's colder, so September or October, they go a whole lot more often, over 100 times in a day. And then if you pay attention to that dotted red line or the full red line here, which represent the median and the 95% percentile, what it shows you is that there's a lot of variation between individuals. And then so obviously we did the same thing for the hippolimnion, and we found that the fish did go a lot more into the epilimnion with about 80, 90 entry into the layer when it's hot, while when it's colder, they go a lot less, about between 20 and 0 times in a day, with, again, similarly to the other graph, we have the 95 percentile, which shows that there is also a lot of variation between individuals. And so here the question became, do the fish go into the epilimnion when it's cool and they go into the hippolimnion when it's hot? Or do we have two behaviors that are actually not replacing each other? So in order to figure that out, we decided to try to group our fish with a PCA based on metrics related to their behavior, which I'm not going to read out loud because I don't have the time. But basically, if I show you the PCA, which explains about 50% of the variation in individual behavior, we're going to find that everything that is on the left side, yes, is related to entering or staying into the epilimnion, while anything that is on the right side is going to be related to entering into the hippolimnion. And so what this means is that those are not two behaviors that compensate each other. It's not they're going to one layer when it's hot and the other when it's cool. This is telling me I have individuals that will go into one layer more and the other individuals that will favor the other layer. And so when we decided to look into the daily pattern, when throughout the day do they go into each layer, we split them into a warm tactic and a cool tactic. Basically, whether they prefer going to the epilimnion, that's going to be the warm tactic, or whether they prefer going to the cool, if they prefer going to the hippolimnion, then they're going to be the cool tactic. And so you're going to have on the x-axis the hour of the day. So we start at midnight and we end at midnight. And then on the y-axis, you're going to have the probability of the fish entering the layer. So in red, the epilimnion, in blue, the hippolimnion. This is based on a study or how-to article by Anna Riley that's based in Constance, which is very interesting to do. If this is the kind of study you want to do, I really recommend that article. And so what we were able to see is that for the warm tactic fish, they are going into the epilimnion around dusk. So those are going to be dawn and dusk, black lines. So they will go with a probability of around 0.5 into the epilimnion, while they go to the hippolimnion a bit before dawn and around dusk again, with higher probabilities in the morning, lower probabilities in the evening. And then when we compare it to the cool tactics, which are the ones that are going to favor the epolimnin, we both find something very different and very similar. So it's similar because the timings are pretty much the same. We're still going into the epolimnin at dawn and at dusk, and we're still going to the epilimnin at dusk. But the difference are the probability of going there. We basically have a probability of 1 of going to the epolimnin, while the probability of going to the epilimnin is down to 10%. So that's a huge shift. And just so to summarize basically everything I've told you today, we were able to quantify the use of the epilimnin. We confirmed that they, all right, well, that wasn't supposed to look like this, but we did confirm that with rising surface temperature, the fish went less into the epilimnin. But for the epolimnin, the usage of that layer did change. It was not constrained by low oxygen. I did not present those results today, but it is available in my preprint. And for the patterns of activity, so when do they go? They did go at dusk when food is more available for the epilimnin. For the epolimnin, the less predictable patterns that we assumed would be found were not found. So we do have that very interesting patterns of dawn and dusk, which we think is also related to food availability. And again, for the last part, you're going to have to come back later today to discover that. And do I have the time? No, I don't have the time. So future research, you will have to come again for the poster. And thank you for your attention. Any questions?
Thank you for the presentation. Really, really cool data set, and the tracking of the fish were really nice. So I was wondering about these two tactics, of how basically they use the different layer, if they're more like the warm or the cooler one. Is this related also to some kind of a physiological response the individual have in their abilities to regulate their temperature faster or slower? Could you measure this?
So we don't know because we haven't been able to measure it. Those fish are dead because it's like 2017, 2018. So we cannot measure it any longer. We do have plans to look into it by basically, we're going to fit a model throughout every single forays that they do to see how long do they stay and like how quickly do they reach the temperature of the layer. Because since a fish is going to adjust to the temperature by diffusion, basically, if the fish is bigger, it's less like it's going to take longer for the fish to warm up, so it should be able to stay longer in that layer. But we did not, like, we looked into the weight of the fish to check if it was related to the cool or warm temperature. It was not the case. And because this had been seen before, we had seen in this specific lake that we had two different tactics. They were called pelagic and littoral tactics before, but now that we have the 3D positioning, we were able to look deeper into this, and we think it's more related to epolimnin and epilimnin use. So we don't know yet, but we'll be diving into this with a heat exchange model.
Thanks.
Hi, thank you for the talk. I was just wondering, so I'm just really impressed by this sensor that can just be tagged to the fish and go everywhere. Is there a limit to the water temperature or anything to these sensors, or was there a time that it didn't work and certain times that you had to control the environment?
I think it worked for most of the time. There are a couple of days where we don't have the temperature of the lakes because the battery ran out, but that's like 3 days in total for a 2 years period. And so those days we simply removed them. Also, they were at the end of the summer or at the very start of the study where we hadn't released the fish yet. So it didn't cause any issues. And for the tags that are inside the fish that give us the internal temperature of the fish, it gives us that information about every minute. There is obviously some loss of information because the tags emit information that if it crashes with other tags emitting that same different information, you might lose some. So there are brief periods where we don't have the temperature or the depth of the fish, but that would be like a 5-minute period most of the time. So if we look at like the hour, within an hour, how much the temperature of the fish changes, or if we're looking within a foray, we can just remove those moments where we don't have the information. So it's not causing any issue because we have so much data. Because again, it's, I actually don't know if I said it out loud, but we have the information, the positioning down to the minute for the fish for about 2 years. So that's a lot of lines. So if we're losing a few, it's really no big issue.
Okay, as we are running a bit late, this will be our last questions. Thanks again for the talk and for the questions. For the second talk of the morning, we can welcome Elena Norman. Elena is a PhD student at the University of Glasgow in the Killen Lab. Is that it? And you will talk about the effect of temperature, again temperature, and sleep disruption on sociality and metabolism on the Trinidadian guppy, which name is Poecilia reticulata. Is that the correct name? Okay, so I think everything is set up, so I will let you the stage. You have a microphone there.
Hi, everyone. I'm really excited to be here speaking to you all today about how sleep disturbance and temperature affect waking sociability and metabolism in the Trinidadian guppy. And this is an experiment that I've just done just this summer, so I don't have full results, but I'm excited to share some preliminary results with you. So a bit of background on sleep. It's a neurophysiological and behavioral state, and it's been found in pretty much every animal that we've studied, all the way from jellyfish and flatworms up to, vertebrates like fish, birds, mammals. And it's near ubiquitous across the whole animal kingdom. But why do we study sleep in animals? Well, it has a huge ecological relevance. So when you have a disturbed night of sleep, it can have implications like a decline in physiological performance, metabolic dysregulation, and also cognitive impairments. And we can, if we have a bad night of sleep, we can have some coffee or something, feel better in the morning. But for fish, they're not so lucky, and they can instead have fitness costs as a result of this. And these can be things like problems with avoiding predators, problems with sociability and living in groups, and also problems with foraging and finding food. And there isn't actually a whole lot that we know about sleep in fish, and there are huge knowledge gaps. And these include what factors might affect sleep, what the consequences are of sleep disruptions, and how climate change could be impacting sleep behavior, and particularly how, and this was covered really well in the last talk, how temperature could play a role in this. But why do we even think that temperature is important in sleep? Well, again, this was covered so great in the last talk. A lot of fish display a temperature preference rhythm, so they behaviorally thermoregulate, and they move from, often in the day, this is for diurnal fish, often in the day they seek out warmer temperatures, and then in the night they seek out cooler temperatures, and we think that this might help their sleep. And we can see, this just shows two different species, the Nile tilapia and zebrafish. Don't worry too much about it, but it just shows a really nice pattern, a sinusoidal pattern that is, they very predictably seek out warmer temperatures in the day and cooler temperatures at night. But this leads us to what happens when the temperatures are heating up and what could be happening to their sleep. So in this study, I wanted to look at how temperature affects sleep, how sleep and different changes in sleep behavior affect sociability and metabolism, and how changes in sleep with changes in temperature affect sociability and metabolism. But because this is a behavioral session, we're gonna not worry about metabolism. That's too much for today. So what I did is I set up 2 different treatments, and I acclimated my fish at 28 degrees or 24 degrees for about a month prior to starting experiments. And then I used 3 different disruption treatments. So either no disturbance, they got to sleep through the night, very happy fish, nighttime or sleep disturbance, and an equivalent daytime or waking disturbance. And the way that I disturbed them is I had, they were in these little custom-built sleep boxes, and there was a pump just outside that would intermittently switch on throughout the day or the night, depending on which treatment they had. And it looks a little something like this. You can see there's some fish that are sleeping, and the pump is about to switch on. Yeah, so to collect the data, I looked at fish using infrared cameras at night to score their sleep using Campbell and Tobler's 1986 definition of sleep, which I'm happy to talk. Oh, there we go. Pumps on. They're not sleeping. Yeah, I'm happy to elaborate on that at another point. But I looked at their latencies to sleep, their fragmentation of sleep. So that's how often they're waking up and going to sleep again and for how long, and also their total sleep duration. And I also then looked at shoaling tendency. So this shows, it's a very common assay in fish, and it's a top-down view. And we use it to basically see how social fish are, so how much they want to hang out with their friends. And because guppies are very social generally, I did a slightly different version of a shoaling assay where they could choose between lots of friends or only a couple of friends. And I tracked where they went using a program called EthoVision, and I looked at metrics like time spent with the big shoal versus the little shoal, how many transitions they did, and like movement metrics like speed and acceleration and distance traveled. And then I also looked at metabolism. I looked at maximum metabolic rate, standard metabolic rate, anaerobic scope. Again, I'm not really talking about that today, but if you want to talk more about that, you can find me at the coffee break. Yeah, so these are some preliminary results. I haven't looked at everything in detail yet. It's a lot of videos of fish sleeping and not sleeping, and it takes some time. But so far, so this is just control fish. So these guys haven't been disturbed at all. So this is just looking at how temperature affects sleep. And you can see that across the night, there is some differences in the amount that they are sleeping. And this is the really interesting bit here, where fish at the higher temperature in purple are sleeping a lot less than fish at the lower temperature in green. And this alludes to maybe a higher sleep latency, so they're taking a lot longer to get to sleep. And again, if we look at state transitions across the night, we can see that this is really the most interesting part. In the middle of the night, there's a huge difference between fish at the lower temperature, where they are waking up more, than fish at the higher temperature, where they're waking up less. But if you remember the last slide, there isn't much of a difference in their sleep duration. So it's probably a difference in sleep architecture, where fish at the lower temperature are waking up more, but just for a short amount of time, and fish at the high temperature are waking up less, but they are staying awake for longer. So if we look now at the sociability metrics that I got from my sociability assays, so this is the total distance moved. There's 24 degrees on the left and 28 degrees on the right. And if we just look at the control and day disturbance fish, you can see that there's a bit of a difference with temperature. And then if we look at the night disturbance fish, you can see that there is actually more distance moved when they've been disrupted at night compared to the day and control fish. But then when we look at it at the higher temperature, this effect is kind of dampened, and there isn't as much of a difference between the 3 treatments. If we look at the shoaling tendency score now, so I calculated this by doing a PCA. So this is a mixture of the total duration with the big shoal, the mean duration that they were spending each time they visited the big shoal, and also how much time they spent not shoaling at all. And this is a way of just getting a more all-round picture of how sociable they are. So again, if we look at the control and day disturbance versus the night disturbance, at the lower temperature, there is a, not quite significant, but there is a difference where the night disturbance fish were more sociable all round. But again, very interestingly, at the higher temperature, this effect is dampened again, and there isn't much of a difference. And then finally, if we look at the zone transition frequency, so this is how many times they go from the big shoal to the little shoal, or the little shoal to the big shoal. So at 24 degrees, the night disturbance fish compared to the control and day disturbance fish, they are switching zones a lot less. But if you remember from the last slide, they were a lot more sociable. So they're probably seeing fish that they know, go into them and sticking with them. But then once again, if we look at the zone transition frequency at the higher temperature, there isn't as much of an effect compared to at the lower temperature. So what could this all mean? So higher temperatures might cause less sleep, but despite more sleep, fish at lower temperatures showed stronger responses to flow-based sleep disruption. And this could be for a few different reasons, which hopefully I will find more about when I do the rest of my data analysis. But this could be an antagonistic effect of two stressors, or it could be some sort of partial versus full sleep deprivation if fish at higher temperatures are all kind of equally sleep deprived, whether they've been whether they've had the flow treatment or not. Yeah, so that's me. For today, I'd like to acknowledge my lab group and my lovely intern Sophia, who helped with this study, and also the wonderful aquarium techs in my university. Yeah, any questions?
Hi, thank you for your talk. I have a very naive question. How do you know whether the fish are sleeping?
No, it's a very good question. So I use a behavioral definition of sleep that I touched on briefly there, Campbell and Tobler's definition of sleep, which is used in a lot of different taxa, but it's basically a combination of circadian regulated quiescence, a species-specific posture or sleep site. So for guppies, they kind of go to the bottom, as you could see in that video, and stay towards the bottom third of the tank. It's also, what else is it? Oh, they have an increased arousal threshold. So that means that if you provide a stimulus when they're awake, they will, like if I poked you when you're awake, you'd be like, oh, hi. But if I poked you when you were asleep, it would take more of a poke or like longer for you to react to the poke. And it's the same with fish. So I test it by dropping a ball in there when they're awake and asleep. And it's also homeostatically regulated. So if they are losing sleep one night, they have to make up for it the next night.
All right, thank you.
Thank you.
I'm sorry, we take only a last question. Right.
Hi, that was super interesting. I loved it. I was wondering for the shoaling behavior, did you also try to measure that at like a basic range so that you didn't have like the influence of the personality of the fish, or is that like a worry for this kind of fish?
It's a really good question. So are you asking if like personality would affect it? You do get more and less social fish, which can sort of affect it. And one way that you can get around it is if you test them before sleep disruption, after sleep disruption, but then you have to eventually like double your sample size. So I went for having a big enough sample size of fish. I didn't put it on there, but I had 180 fish in there, so that if there was variation in their individual like personality and how sociable they were, then hopefully within like between treatments you could still see differences despite the variation, if that makes sense. Thank you.
Okay, I'm sorry I have to get short questions for you, but don't hesitate. Go and ask her at the coffee break. And well, for the last talk before it's coffee break, we can welcome Léo Maucourt. Léo, you are a postdoc student at the University of Antilles in the Laboratoire de Biologie des Organismes, des écosystèmes aquatiques. And you will talk about the description of the behavioral context of underwater sound production in juvenile green turtles, Chelonia midas. So, stage is yours.
Thank you. Hi, everybody. I'm Léo Maucourt, and I'm about to present you a part of the result of my thesis about the context of sound production underwater in green turtle Chelonia midas. So, first of all, why we should be interested in the sound production in sea turtle? We know the understanding of sensory ecology of animals, that is how they perceived and respond to the environment, could be an effective tool for the conservation of endangered species, such like sea turtle. Some studies show that there is some sound production underwater in freshwater turtles, and some results suggest it could be acoustic communication. However, in sea turtle, we had no information about their production underwater. That's why we published a first article describing the sound repertoire of green turtle underwater. However, we did not have enough information to conclude about any acoustic communication, and it was one aim of this study. We also wanted to understand the context of production to effectively improve the sea turtle conservation at lower cost and effort. So our data collection took place in the Caribbean Sea, in Martinique, and more precisely at Léance d'Arlette. So we tagged 23 green turtle with a biologger, which have many advantages. So, for example, biologger could contain many type of sensor, for example, camera, hydrophone, accelerometer, etc. And all these sensor allow us to have a data collection following the focal animal survey method, consisting into the tracking of an animals to record all the states and events over the time. This type of biologger also allow to reduce the disturbance of a tagged animal thanks to an automatic release system integrated in the device. So this is an example of what we recorded. So here you have the tagged turtle scratching her head against the sponge, and if you hear well, she produces some sound. Okay. So to do my analysis, I use Avisoft Bioacoustic. This is the screen I mainly work on. So in blue, you have the oscillogram with the amplitude over the time. And in red, you have the spectrogram with the frequency over the time. And the variation in amplitude is represented by the variation in color. So the blue is low amplitude and red is high amplitude. I also use a label tool. So I labeled many elements on my recording. So, for example, I labeled the external event, mainly on video and on spectrogram for the sound bot, for example. All sound were labeled directly on the spectrogram, and for the behavior, I just use the result of of an algorithm that automatically identify behavior label thanks to accelerometer data. So in total, I use 11 type of sound, 6 type of behavior, and 3 type of external event. For each of my label, I analyze them, with air routines and demi-coding method. First, to measure the frequency of the event, but also to see the co-occurrence between a sound and a behavior or with an external event. I also did a Monte Carlo simulation to assess the significance of co-occurrences. So for each of my label, I had a start and a stop, so a duration, and then I translated this duration with a demi-coding method consisting into cutting day in 1 second sequences. And for each label, I have a 1 if I have a label and a 0 if I have no label. So here, for example, I have the long squeak that have a duration of 6 seconds, and 2 seconds of the long squeak co-occurred with swimming behavior. So I can say 33% of my long squeak co-occurred with the swimming behavior. So this is the result. My sounds are classified in 5 categories. The first category is the pulses, with the mono, the doublet, the triplet, the multipulse, and the talk. This is an example of the multipulse. As you can see, all these sounds mainly co-occurred with resting and swimming behavior. And the Monte Carlo simulation show that the co-occurrence with resting behavior is significant. The second category is the low amplitude call with the croak and the rumble. Once again, this sound mainly co-occurred with resting and swimming behavior, but one very interesting result is the huge number of sound for the rumble. And all this sound was mainly recorded on 3 tags after sunset. And we had a few clues that indicate there is not only one individual that produce all this sound, but several individuals near to the tagged turtle that produce all this sound. For example, on the little spectrogram here, amplitude of each sound is different, and some sound overlap each other. So it's similar to chorus behavior. This is an example of what we heard. Okay. The third category is the frequency modulated sound. Very special. This sound mainly occurred with scratching behavior. That is a bit difficult to interpret. The 4th category is the squeak, with the short squeak and the long squeak. Once again, this sound mainly occurred with resting and swimming behavior, but for the long squeak, also significantly co-occurred with the presence of human in the camera field, and sometimes during a flight behavior of the turtle. I will not have the time for the video, sorry, maybe later during the question. During question, if you want. The 5th category is the grunt. Once again, this sound mainly co-occurred with resting and swimming, but it also significantly co-occurred with the presence of conspecific in the camera field. It's also the only one sound for which we have a visual clue of the sound production. So this is a short example. Here's a tagged individual producing the sound with the little movement of head retreat during she approaching conspecific. So to finish, on the 11 sound, we identify 4 of them in a specific context. For the FMS during scratching, we suggest it could be linked to an emotion with positive valence. Long squeak could maybe linked to an alert behavior. We have to explore it. The grunt could maybe linked to warning or food competition behavior. And for the rumble, I'm writing a paper right now, and in this paper, it will be published soon. I suggest it could be linked to social cohesion or collective anti-predator behavior. Maybe I will speak about that next year. And after this study, so we need to go further to understand the biological role of this sound, and not only for the 4, but only for the rest of the sound repertoire of the turtle. We also had to compare the juvenile sound production with the adult sound production and discover if there is sound production in all of the sea turtle species. Thank you.
Thank you. Wow, it sounds very difficult to make some sense of these songs, but yeah, really good job. My first question is, why do you think they are scratching? Do they have like algae growing on them that they're trying to remove?
Okay, it's a difficult question. I'm not sure of why they're scratching. Maybe, yeah, just because it's to remove some dead cell, or I'm not sure.
It's interesting that they make a specific sound when they are removing something from their body.
Yes, yes. They often scratching their shell against a rock. And I observed they produce this sound during scratching when there is absolutely nothing around. So there is no predators, no other conspecific. So it's why we suggest it could be linked to an emotion with positive valence, because maybe just a hollow case, so it's cool.
Okay. And did you ever see mating? And if they make a sound before mating?
In this population, it's only juvenile sea turtle, so there is no visible mating behavior, and this is why we need to compare the juvenile sound production with adult sound production to understand the sound that could be linked in the mating behavior.
Okay, thank you.
Take the last question. Thank you so much for the wonderful presentation. I have a question on the audio loggers themselves. What software did you use to log in the data and differentiate the different types of, I think, sounds?
I'm sorry, could you repeat?
What software did you use, like application to use to like log in the data set and then differentiate the different sounds?
to identify all sound.
Yes.
It's just me. I don't use any application to this. It's a bit long, but yeah, to discover all the repertoire of the turtle, it's just hour and hour and hour of listening and uh, remember what is the structure of each sound to say, okay, I see a pattern of this type of sound, and after I classified it with my...
Beautiful work, man.
Thank you. Just 10 seconds.
Oh, est-ce qu'on a le temps? Leo, is it that one?
No.
This one? Let's watch the video then.
Okay. So you have which go to breathe at the surface. When she reaches the surface, we can see she's face to land. And when she dives so quickly. Oh, she dives so quickly that the camera partially removed. When we stop, so at the back of the turtle, and we can see three divers behind her. So I suggest she maybe feel stuck between land and diver. And so when she flies diver, we can hear. Maybe you have here the little.
Yeah. We believe you. Thanks. Thanks. Okay, thanks, Leo, for these really cool presentations. I have 3 pieces of information for you. The first one, if you haven't sent your presentations online on the dedicated website, do it as soon as possible. The second one, we're going to do a 15 minutes coffee break. So let's meet there. And the third one, keep your Eco Cup from the coffee break. You will need it in the posters sessions in the afternoon. Let's meet in the coffee break then.
Okay. So now we will welcome Jeanne Legros. She's a PhD student at the Biogeoscience Lab in Dijon.
So
So hello everyone, I'm going to talk to you about the impact of urbanization on the competition between blue tits and grey tits, these two little birds you can see here. So first, a little definition, because I'm going to talk a lot about acoustic niches. So as well as they have ecological niches, organisms that produces sounds also have acoustic niches. So it's a partitioning of the acoustic space in terms of frequency. For example, bats use ultrasounds to communicate, while amphibians use really low frequency songs. But also in terms of temporality, for example, you're most likely to hear birds singing during the day. Most bird songs sing during the day, but bats and amphibians mostly vocalize at night. So they all use different acoustic niches. At a smaller scale, between species inside a taxa, it may happen that some species have really close acoustic niches. It's the case of blue tits and grey tits. So I'm going to diffuse two sounds. I want you to pay really attention to the structure of the song. So this is a blue tit song. And this is a grey tit song. So it's quite similar in structure, and just the blue tit sings a little bit higher than the grey tits. This is what happens when they are in allopatry, but when they are in sympatry, the issue is that they have two similar acoustic niches, so there is an important overlap that produces a high interspecific competition between them. So there have been a study that showed that in sympatry, blue tits will change their song a bit through a process known as character displacement. And so they will add a trill at the end of the song. A trill is a note that is repeated very rapidly. So this is the kind of songs they emitted in sympatry with grey tits. So it's slightly different. Many environmental aspects can decrease the acoustic niches that is that's available for organisms to use, and one in particular is the urban environment. And because urban noise has two main characteristics. The first one, it's discontinuous. You have really busy hours during the day, in the morning and in the afternoon, and it's pretty quiet at night. So some birds shift their acoustic niche and sing at night. It's the case of robins in New York, for example. When it's noisy areas, they sing at night to escape the sound during the day. The other characteristic of urban noise is that it occupies low frequencies. The issue is that many organisms use these frequencies to communicate, and so there is a risk that their signals are going to be masked. So many bird species have been shown to increase their song frequency in response to noise. So here we wondered, how does urbanization impact interspecific competition between these two species? Because indeed, if grey tits increases its song frequency, the overlap with the blue tits acoustic niches is going to increase, and then perhaps the competition is going to increase. And if it does, how is the blue tit going to respond? Is she going to sing even higher? So that was the first study we did between 2022 and 2024. We recorded birds, we measured different frequency parameters, and we found out that indeed grey tits increase the maximum frequency of the song in response to noise, so to sing above the noise. And they also decrease their minimum frequency when there is a lot of blue tits around, so when the interspecific competition is high, while blue tits don't change their song at all, nor in response to noise, nor in response to competition with grey tits. So this is what we had, grey tits singing in a more important frequency range. So we have the overlap that increases here because they increase their maximum frequency. So we wonder, maybe the blue tits doesn't change their song, but maybe they react differently to this kind of songs that have a higher maximum frequency, so a bigger overlap. And we also wondered, why does the grey tits decrease the minimum frequency of the song when there is a lot of blue tits? Does it change the territorial response of
blue tits. So that's what we tested this year. We did paybacks experiments in Dijon and Besançon. So we focused on blue tits and we put speakers in their territory during the reproductive period. And we diffused different types of songs. So we had two different tests for each question. First, for the impact of noise, we diffused great songs that were emitted in a silent context, so with a high maximum frequency. And we diffused also graded songs that were emitted in the noisy areas, so with a high overlap with the Bluetooth song. We used Bluetooth songs as control. And then for the second test, where we tested the impact of competition, we exposed Bluetooth to graded songs emitted in the context of no competition, so with high minimum frequency, and graded songs that modify the song in response to the competition to see if it decreases the response of Bluetooth. We focused on different behaviors, the number of vocalization, the approach distance, the latency to react, and the duration of vocalization. We used the hurdle model because we had a lot of zeros, so it helped us deal with that. So first, I'm going to introduce the result for the impact of noise. So here you can see that the approach distance, we didn't have a significant difference between the two graded song treatments, but there is a difference with the control. So Bluetooth can discriminate if it's a graded song or a Bluetooth song. But we didn't find any difference for the number of vocalizations. So they vocalized equally in all three treatments. Then for the impact of competition, we still use hurdle model. So first, for the approach distance, we found the same result as for the impact of noise. So no significant difference between the two rated song treatments and a difference with the control. But this time, for the number of vocalization, we found that there was a significant difference between the control and the high abundance, the high repeated abundance treatments. So when exposed to songs that graded emitted in the context of high competition, they vocalized significantly less. So this showed that when graded increase their decrease their minimum frequency in response to high abundance of Bluetooth, it helped decreases the territorial response of Bluetooth. So it's a way to decrease the interspecific competition between them. However, even if graded songs increase the maximum frequency, which increases the overlap between their niches, it doesn't change the territorial response of Bluetooth. So maybe the overlap is not that important. Maybe the increase in frequency is not important enough to really increase the competition between them. Maybe because Bluetooth have now a different structure, a different song structure, because they add the trill at the end of the song. They have different enough structure in songs, so the competition is not so important. So this is something we have to investigate. In this study, we focused only on parameters of the song that are frequency and duration, fine parameters. But a study in 2008 showed that actually the shift in frequency is not necessarily linked to just singing the same song types higher. It's actually changing the song types that are already higher. So we also need to investigate if some song types are more competitive from others, because maybe more similar to Bluetooth song or something. And that's all. Thank you.
Thank you for the great talk. Do you know whether or not it has already been investigated if other characteristic of the landscape has an effect on competition between blue and gray tits?
No. I know that they showed that when there is a high density of blue tits, gray tits decrease the number of eggs. So there is a competition. They also compete for food. So I guess when it's like a really dry summer or something, they will have less food, and so the competition will increase. They compete for almost everything because they are really close ecologically, like they nest in the same thing. So yeah, there are studies about the use of nest boxes. For example, great tits are more easily investigating nest boxes than blue tits, but yeah.
Hi, I just have a question. Do you have like some data or some analysis regarding like, for example, long-term consequences of this increase of frequencies in great hits?
No, we actually don't know if it's a long-term change or if it's just like in the moment. This is something we want to investigate because when we recorded them, we also had a microphone that recorded the environments, and we want to see if it's something that they do, like while a car is coming, they would sing in higher frequency, or if it's something more long-term. We don't know for now.
Okay, thanks.
Maybe one more question?
Hi, thank you for your talk. When your treatment had the call of the great tit with urban or not much urban noise, how did you manage to disentangle between the effect of the noise itself and the call in response to the noise?
Sorry, can you repeat the end?
How do you know if it's the effect of the call of the song or the effect of the urban noise in the recording?
We filtered the urban noise.
Okay,
so it's just
you remove it.
Yeah.
Okay,
So there is no background noise in our treatments.
that makes sense. Thanks.
Yeah.
Okay, so I welcome now Jikang Park. He's a PhD student here at the CEF in Montpellier.
Thank you. So today I will explain about my preliminary result about the sleep of the southern elephant seals on the land. Okay, so we can find many interesting behavior from animals' active state, like the flying or migration, or the foraging behavior, predator avoidance, or sometimes mating. But also, we can find very important and interesting behavior during the inactive state, the sleeping. So what is the function of the sleep? So many people think the main function of sleep is just the energy conservation, but usually during sleeping, they conserve a very small amount of energy. So then why animals are asleep? So there are many important functions about the sleep. So sleep can support the restorative and cognitive and developmental processes. So sleep is very important for animals. This is why all of the animals from the mammal species to small invertebrate species, all of these animals are asleep. So it's the same in the marine mammal species. So I interested in the pinniped, the marine mammal species. So we can find many interesting sleep strategies from the marine mammals. So they can sleep on the land, but in the ocean, they have to sleep. So they want to escape from their predator. So for example, the otaride, one of the species of the fossil, can perform the unihemispheric sleep. It means that they can sleep with the half of their brain. At the same time, the other part of the brain is awake, so they can monitor their shallow depth predator. And also we can find another interesting strategies from the fossil, the true seals. So here is another study about the northern elephant seals. So according to this paper, they can sleep in the depths briefly. So like this, we already know about many interesting strategies from the marine mammal species, but we don't know, still we don't know many things. Like for example, according to this paper, northern elephant seals are usually sleep around 10 or 11 hours on the land, but with this strategy in the ocean, they only sleep around 2 hours per day. So it's very short duration. So how can they manage their sleep? To understand the whole life sleep from the animals, we have to study sleep for the long duration. So then how can we know their sleep? So here is my study species. So my study species is different species. The southern elephant seals, they live in the Antarctic Ocean. So usually they spend most of their time in the ocean. For example, after finishing the molting period, they go back to the ocean and they spend the longer than 6 or 7 months in the ocean. In that time, they can obtain a lot of energy from the fish, their prey, but it's hard to sleep because it's very cold and they need to breathing every time. On the other hand, on the land, they can sleep well. For example, during the molting season, they spend almost a month on the land. And on the land, there's no food and no predator in the Antarctica, so they don't need to be an active state. It means they can sleep as much as they want with the inactive state. So then, how can we know their sleep and inactive state? So here is another video of the elephant seals. So do you think this individual is now sleeping? Do you think? So this question is about, do you think the inactive state can be a good sleep indicator for southern elephant seals? So to know this, we need to understand about the inactive state of elephant seals and also real sleep state of these species. So at first, I prepared 2 types of the experiments like this. So I installed several video camcorder in front of the seals. So I collected video data from them. And also I deployed several data logger with the accelerometer, gyroscope, magnetometer, and also the temperature sensors. So we deployed 14 devices on the animals. And also, so we only retrieved 9 devices because we used 5. But anyway, we obtained totally the 21 days of movement data from 9 individuals. So based on this data set, from the video recording data, we can identify their active state. So if they move their neck or if they scratch their body, I checked it as the active state. And it isn't, I checked it as the inactive state. And also we have the movement data based on the accelerometer and gyroscope. And also we have the paired video recording data. So we can make a good threshold to identify active state. And also we have the temperature data. So we can know the animals in the ocean or on the land based on the temperature. So now we prepared everything about inactive state. So here, so the upper part is about the video recording data, and the below one is from the movement data from the accelerometer. So it looks a little bit different because based on the video recording data, we can identify their small movement like that they are scratching. But if the animal is moving a lot, so if they are going out from the video frame, we cannot follow the animal's movement. So usually the result of from the video recording data can be underestimated. But anyway, we can know that usually they are very inactive state on the land, so just less than 15%. So now the main question is this. So how can we know their real sleep? So to understand this, we prepared a slim logger. So it's very different with the active or inactive ratio. So we collected the EEG signal from the animals. It sounds very difficult. The full name is the electroencephalogram. It's very difficult, but very simple. It's the kind of brain activity data which can identify the sleep. So if we want to obtain the EEG data, we need to place the electrodes near their brain. So like this, there are several methods, like the upper one. So on the northern elephant seals, they use the surface mounted method. It's very non-invasive method, so it can be very good for animals, but it's hard to obtain the clear, strong signal, and also it's not fit for long-term recording. So that is why we choose the different method, the minimally invasive subcutaneous method. So we use very long electrodes and insert this under their skin, and then we can obtain very clear, stable EEG signal from animals. So signal looks like this. So how can we identify sleep based on this? So if the animal is waking, the signal looks like very low amplitude and high frequency. But if the animal is going to the sleep, the most of the neuron activity in the frontal area of brain are synchronized, so we can get very the slow wave and high amplitude signal. We call it as the slow wave sleep, and usually the frequency is from 0.5 to 4 hertz. So the two of the picture looks very same. I know it's very same, but left one is during waking, and right one is during sleeping. So we can identify sleep based on the EEG signal. So again, we can make some good threshold to identify real sleep state. And then here's the result. So we already checked the first graph, the pie chart and the hourly active ratio. So their inactive ratio on the land is around 85%, but the slow wave sleep ratio is less than our expectation. It's less than 50%. And if we only pick up the inactive ratio, the 85%, the slow wave sleep ratio is only 56%. So it means inactive state is not good sleep indicator for the southern elephant shears. So the conclusion is like this. So according to the previous study in the ocean, so the animal can sleep with a very, very strange strategy. So they can sleep more than our think. And also on the land, even if they are with a very long duration of the inactive state, usually they can sleep less than our expectation based on the EEG method. So if you want to understand about sleep in the wild individuals, so it depends on the species, but for the marine mammal species, you have to use the EEG method. So this is my take-home message. So thank you very much, and if you have any questions, feel free to ask me. Thank you.
Maybe I missed it, but in the last video you showed, we see the breathing, and I was wondering whether, like, from the video of the breathing and the rhythm of the breathing, if that would be a even less invasive proxy that would work to identify sleep.
Yeah, it can be useful, but it depends on the species. So in this preliminary result, I only show the slow wave sleep. But as you know, the sleep has the different level. So like there is the REM sleep and non-REM sleep. We have the different types of sleep. So if we want to know all of the types of sleep, we really need the EEG method. So for example, for this individual, we deployed the devices on their head. In that case, it's really hard to get their breathing signal.
Okay.
Because it's on here.
No, but from the video, you
you
can see the breathing.
And also with the video, so it's the good view of the video, so we can know some breathing rhythm from here, but it depends on the videos, the quality and the position of the video. So sometimes it's hard to identify their breathing rate or sleeping, the inactive ratio.
And do your devices, do they work underwater? This device you described that are not too invasive, could you use the same one to measure the sleep in the water, or it's different?
So many people think it can be very invasive method, but we use the painkiller and many drugs to clean here, and then we insert here. Okay, so we insert the electrodes from there, and then we clean that site, and then we deploy the device on their injury site. So I think it's okay for them because the device and the many glues can shield their injury site. So I think it's okay for them. There's no any negative effect on the individuals until now. So I think it's okay.
Undo? Okay. Thank you. It was really interesting. And I was wondering, do you observe differences between male and female in the sleep?
A very good question, but we only focus on the female individuals. This is because, so this is the wild animal study. So in here, in front of the other shares, this individual is a female. And this is usually the 300 or 400 kilogram, and it's bigger than 2 meters. It means the behind individual is male. They are more than 2 tons, so it's really hard to deploy some devices on them. So usually many scientists only focus on the female for them. But later, maybe I want to study about the male individuals. For just now, we only focus on the female individuals. Thank you.
Okay, now we welcome Anaïs Coton. She's a PhD student at the Centre de recherche sur la biodiversité et l'environnement in Toulouse, and also in the Institut pluridisciplinaire Hubert Curien at Strasbourg.
Thank you. So hello everyone, I'm glad to be here. So today I'm going to present you a part of my ongoing thesis project. This presentation is called Hormonal Trade-off in Parental Investments, Effect of Corticosteroid on Prolactin and Behavior in King Penguins. So reproduction is one of the major components contributing to species fitness, and parents need to dedicate time and energy to feed their chicks and to ensure their success and survival. Therefore, parents are exposed to quite a delicate balance between either investing this energy and time to their current offsprings or to keep this energy and time to ensure their own survival and then contribute to future offsprings. And how they manage this balance is reflected in what we call parental investment. So parental investments is defined as any cost or expenditure that are associated with raising the offspring, ensuring that the offspring survive and ensuring the offspring's reproductive success, but also at the same time, this reduces the parent's ability to invest in future offsprings. And we know that it's both shaped by ecological conditions like food availability or predation risk and the parents' physical mechanisms. And both of them affect the allocation of resources. And we know that a way that both these factors affect the allocation of resources is through hormones. So hormones are molecules that are continually produced within one body. And we know that they act as mediator between environmental cue and parental state, as well as between environmental cue and parental behavior. They also act as mediator between the parental physical state and the parental behavior. And in birds, there are two main hormones involved in parental investments. The first one is the prolactin hormone, also called the parental care hormone, which promotes offspring survival and reproductive success. And the second one is the corticosteroid hormone, known as the stress hormone, which promotes parent survival and future reproductive events. So I'm not going to detail there, but various two hormones are synthesized by two different pathways that may interact with one another depending on the context. In a case when the individual is exposed to a short-time stress, the production of corticosteroid is associated with an increase in the production of prolactins. But when the individual is exposed to chronic stress, it is quite the opposite, with the production of corticosteroid being associated to a decrease in the prolactins. But again, this relationship is highly context and species dependent. So the king penguin is a subantarctic avian species that has a long reproductive cycle on land that can span over a year. They are exposed to strong environmental constraints during breeding, which are likely to raise their corticosteroid level. Despite living in such strong environmental, environmental constraints, they show quite a peculiar response to a short-time stress.
they are upregulating their defenses. However, the effect, we don't know if it's they might present adaptive mechanisms to chronically elevated corticosteroid or stress hormones level, and we especially don't know the relationship between the corticosteroid and prolactin, and the effect of this corticosteroid on their behavior, and particularly on the parental investments. So to investigate that, between 2018 and 2019, adult king penguins were implanted with corticosteroid implants, which release the hormone for about 19 days. And other individuals were only implanted with the implants, but without the hormones. And only one individual per pairs were implanted. And we monitored them at 4 time points, D0 corresponding to before implantation, D6, 6 days after implantation, and S1 and S2 corresponding to capture that we made after they did the first foraging trip or a second foraging trips, and we monitored both the hormones levels, so prolactin and corticosteroid, and the conditions. Then we conducted during the whole breeding cycle short and punctual behavioral session, looking at behavior punctually for about 30 minutes. We recorded 4 behavior every minute. And then we also noted the times that they spent at sea. And then we looked at the chicks. So in this study, I only include the chicks' weight and the chicks' survival through this breeding cycle. And I'm not going to go into detail for the statistics because it's a bit complicated, but you can ask me a question later. So for the results, first for the corticosteroid hormones, you have in purple the coach implanted individual, and we show that at both day 6 and S1, this individual had a higher baseline corticosteroid level, but it was not the case at S2. And then for the prolactin, we show that at each time point, coach implanted individual had a lower prolactin hormone level. For the body condition, we show that at both incubation and during chick rearing, coach implanted individual showed a lower body conditions. Then for the behavior, we showed 2 different patterns. So during incubation, we had lower vigilance and aggressive behavior. Wait. So you have the color if you need. And it was quite the opposite in chick rearing, where we had higher vigilance and aggressive behaviors. So for the time spent at sea, we show that during incubation, the coach-implanted individual spent less time at sea. And then when we look at the chicks, we saw that starting at day 35, a chick having 1 parent implanted with stress hormones had a lower weight than one with just the implants. And this difference increased in time because between day 35 and more, you have a height chicks mortality. And talking about chicks mortality, we show that chicks from the coach implanted groups had a lower survival probability than one from the control groups. So then what we showed is that the artificial elevation in corticosteroid was associated with a lower prolactin level in these individuals. However, as the corticosteroid level in this coach-implanted individual did not differ at S2, but that prolactin was still lower in this individual, it is likely that the effect of the corticosteroid on the prolactin was indirect. And we showed that previously that the increase in corticosteroid leads to lower individual body conditions. And we know that having lower body condition is likely affecting the production of prolactin. So it is likely that this was through this pathway. And then we know that both hormones influence the individual behavior. So now I'm going to talk about one because they are each different pathway. So we showed that there was a decrease in the time spent at sea for coach-implanted individual. Normally, individuals spent around 14 days during the incubation period at sea, which is the most important period in which they can accumulate resources before the egg hatch and before the energetic demand increase. And we know that for all individuals, we were at about 10 days. And we know that losing three or 4 days is equivalent to losing 6 kilograms of resources at sea. So it is likely that this decrease in the time spent at sea was associated with a decrease in foraging efficiency, which likely affected back the individual conditions. And this decrease in foraging efficiency likely affected the quantity of resources that the parents could give to the chicks, which contributed to a lower chicks weight before winter. So day 35 is right before winter, and starting at winter, chicks can fast for up to 4 months. So we know that in this species, if you are below a certain weight, your survival is null. So it is likely that because of that, we had a lower chicks survival. So to conclude, this artificial elevation in corticosterone hormone lead to a change in parental investment, and more especially this balance favors the parents' own survival and self-maintenance, and maybe future reproductive events, which can be quite concerning at the long term because we know that they are more and more exposed to different stressors. And I wanted to finish by saying thank you for all of you to be here and for all the people who contributed to the study, because it was quite a long field work and laboratory work too. So yeah, and thank you for listening.
Hi, so thank you for your talk. It was very interesting. I have a question because we clearly see the impacts on the chicks, but I was wondering if you also studied like the cohort or aggressivity levels of the chicks, or if I do not survive enough for you to know that kind of thing.
So here in this study, we only studied the adults, but there was another study made by my supervisor on Wednesday on the effect of corticosterone on the chicks' behavior. So here, chicks' behavior was not recorded, but we recorded the behavior of the adults. And we show that, yeah, there was a less aggressive and vigilant behavior at incubation. And it was quite the opposite when there was chicks. And we know that parents can obtain different signal depending on whether it's an egg or it's a chicks. So yeah, no, in this study, we don't have the behavior of the chicks. Yes.
Hi, it's very interesting topic. I just my question is about the corticosterone because, like, there are two kinds of effects related with corticosterone, that is stress, or also the metabolic rate. So, do you have like any idea if your results can be linked to one or the other, according to the behavior of the chicks, to the chicks?
So you're asking me if either the corticosterone is having a stress effect or the metabolic rate effect?
Or if it's like a consequence of metabolic rate also.
Oh, here it's, we are starting, so here what happened is that we artificially induce first the corticosterone and then we assess, because it's only part of my ongoing thesis, but we assess the effect on the individual metabolisms. And globally, we show that there is a moderate response within the individuals. So, but again, there is there is a decrease in parental investment. So we are most probably that the effect of the stress leads to the chicks being less fed. And then we know that below a certain weight, starting winter, it's mean death for them.
Okay, thank you. It was great talks. So now you will hear my beautiful English to give you some informations. So I have two informations for you.